My aimers, now we will look at the phases of dark reactions. So do remember that dark reaction is divided into three basic phases. First phase is called as carbon fixation. The second phase is called as reduction, and the third phase is called as regeneration of carbon dioxide acceptor. Now we will study all of these steps in detail. So first point is of carbon dioxide fixation. Now what happens in carbon dioxide fixation? The reactant over here is carbon dioxide, 1,5-ribose bisphosphate, and the product is phosphoglyceric acid. Now we require 3 molecules of carbon dioxide. Why do we require? Because the product of dark reactions contain 3 carbons, and that carbon will be from carbon dioxide. So we need 3 carbon dioxide molecules, and as the molecules of carbon dioxide are 3, so the one which will accept the carbon dioxide should also contain 3 molecules. So we have taken 3 molecules of 1,5-ribose bisphosphate, and the product is then 3-phosphoglyceric acid or 3-phosphoglycerate, and the reaction is catalyzed with the help of an enzyme which is called as Rubisco. Now look here, that this carbon dioxide, I am dividing this carbon dioxide into 2 components. Firstly, you see here that all the 3 carbon atoms are added to 1,5-ribose bisphosphate to make phosphoglyceric acid. Now there are 6 oxygens present in carbon dioxide. 3 2s are 6. I have divided these oxygens as 3 oxygen and 3 oxygen. Now these 3 oxygens add to 1,5-ribose bisphosphate. But do remember that these 3 oxygens are still remaining. I'll tell you later that where do these 3 oxygens go, but only 3 oxygens add to ribose bisphosphate. Do remember this point. Now you can see here these are the 3 atoms of carbon dioxide. Okay, 3 carbons are over here, and the structure of 1,5-ribose bisphosphate I explained here. I explained in the last slide that this is 1 molecule of ribose bisphosphate, 5 carbon atoms, and phosphates are present on carbon number 1 and carbon number 5. So I have taken these 3 molecules of ribose bisphosphate. Now carbon dioxide adds to 1,5-ribose bisphosphate, and what happens next? So first remember that these molecules in total these molecules have 15 carbon, 5, 10 and 15. So 3 molecules of ribose bisphosphate have 15 carbon. Now what happens over here is that this molecules of carbon add to ribose bisphosphate. Like this is whole the structure of ribose bisphosphate. Only the carbon number 6 over here, you can see 1 carbon from here, 2nd carbon from here, and 3rd carbon from here adds to the molecule of ribose bisphosphate to make 6 carbon compound. This 6 carbon compound is an unstable compound. It is not stable, so it cannot maintain itself. So what happens that this 6 carbon compound immediately breaks into 2 molecules of 3 carbon compound. Okay, like it breaks from the center. I'm just explaining it. I'm just saying like that only for your understanding. Otherwise, it's a very complex process. So when it will break from the center, then what will be the product? There will be 2 molecules of phosphoglyceric acid. Do you remember that this was the structure of phosphoglyceric acid? 3 carbons and phosphate was present on 3rd carbon. So the 6 carbon unstable intermediate breaks into 2 molecules of phosphoglyceric acid. Now the carbon which was originally a part of carbon dioxide has now been added into phosphoglyceric acid. You see that this blue carbon, 3rd carbon, is from carbon dioxide. So it means that carbon has been fixed. Also remember one more point, that in your textbook it has written that this 6 carbon unstable intermediate breaks into 2 molecules of phosphoglyceric acid. It is true. But in total these are 6 molecules, like 1, 2, 3, 4, 5, 6. These are the total 6 molecules, but 2 molecules of phosphoglyceric acid. Okay. So in total these are 6 molecules. In diagram it is mentioned 6, while in textbook in the line it is mentioned 2. So these are 2 molecules of phosphoglyceric acid, but in total these are the 6 molecules of carbon. And how many carbon are present over here? You see that 3 carbon here, 3 carbon here, 3 carbon here, 9 and 9 are here. So total for 2 molecules of phosphoglyceric acid in total contains 18 carbon atoms. Got it? So carbon dioxide adds to 1,5-ribulose bisphosphate to make phosphoglyceric acid. Now the next step is reduction. Reduction means addition of hydrogen, okay, or the addition of electrons. Now what happens in the step of reduction? That the product which was formed, this 3-phosphoglyceric acid, and we saw that total were 6 molecules, it adds 6 inorganic phosphate into it, and the product is then 1,3-bisphosphoglycerate. This reaction takes place in the presence of ATP. As there are 6 molecules of phosphoglyceric acid or 3-phosphoglycerate, then 6 ATP are used in total. So 6 molecules, so 6 ATP are used. Now how does the process occur? You see that I have 6 molecules of this 3-phosphoglyceric acid, 1, 2, 3, 4, 5, 6. Now what happens? That the 6 phosphates are taken from this 6 ATP molecules. These ATP are those which were synthesized in the light reactions. So these ATP break to make 6 ADP and 6 inorganic phosphates. So these 6 inorganic phosphates add to this 3-phosphoglyceric acid. Okay, and the product over here is 1,3-bisphosphoglycerate. Like all of these phosphates are then attached to the carbon number 1 of phosphoglyceric acid. You see here that carbon number 1 of phosphoglyceric acid, so it is called as 1,3-bisphosphoglycerate. Got it? And how many carbons are present in 1,3-bisphosphoglycerate? 18 carbons are present in 1,3-bisphosphoglycerate. You see 3, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Got it? And the product is 1,3-bisphosphoglycerate. So it is reduction is basically the step where the 6 ATP molecules are used. Now, what is the next process in the reduction? The next process is that 1,3-bisphosphoglycerate now adds NADPH into it to make glyceraldehyde 3-phosphate. Now, do remember that 6 molecules of 1,3-bisphosphoglyceric acid were used. So, we use 6 molecules of NADPH. And similarly, 6 molecules of G3P are formed, and these 6 NADP make 6 NADP plus and donates the inorganic phosphates, and it is also the step where the water molecules are produced. We will see how the process occurs. So see here we have 6 NADPH over here which were formed in the light reactions. These 6 NADPH donates 6 hydrogen and 6 electrons. Got it? So what happens that this is the structure of 1,3-bisphosphoglycerate. Carbon number 1 and carbon number 3 contains 2 phosphate, and these are the 6 molecules. And then the electrons from the NADPH adds to the 1,3-bisphosphoglycerate. Okay, and the product over here is glyceraldehyde 3-phosphate. So the addition of electrons is called as reduction. So this 1,3-bisphosphoglyceric acid basically undergoes reduction to make glyceraldehyde 3-phosphate. And all these phosphates from carbon number 1, okay, this is carbon number 1, all these phosphates are removed. Remember, whenever there is addition of electrons, there is always the loss of phosphate groups. So these phosphates, do remember the phosphates from carbon number 1 are removed. You don't have to remove it from carbon number 3 because the product is glyceraldehyde 3-phosphate. It means glyceraldehyde 3-phosphate contains phosphate on 3rd carbon. Okay, so phosphates are removed from the carbon number 1. Phosphates are present on carbon number 3, so it is called as glyceraldehyde 3-phosphate. All these 6 phosphates are released here, and the product is G3P, which contains 18 carbon atoms. You can count from here. So now these 6 electrons have been used. They have been added into 1,3-bisphosphoglyceric acid to make glyceraldehyde 3-phosphate. These 6 hydrogens from 6 NADPH are remaining. Now what happens to this 6 hydrogen? That I can write these 6 hydrogens as 3H2. Okay, and I told you in carbon dioxide that 3 oxygen are remaining in carbon dioxide. Now that 3 oxygen from carbon dioxide adds to 3H2 to make 3H2O. So do remember that 3 molecules of water are produced in the process of dark reactions. The hydrogen in the water is from NADPH and the oxygen from the water is by carbon dioxide. Got it? So this was the second step, which was reduction. So do remember that addition of electrons occurs in 1,3-bisphosphoglycerate, that is why it is called as reduction. Addition of hydrogen occurs in oxygen, that is why it is called as reduction. And do remember one MCQs, that this glyceraldehyde 3-phosphate or phosphoglyceraldehyde is formed as a result of dephosphorylation. Dephosphorylation means removal of phosphate groups. So phosphate groups are removed from 1,3-bisphosphoglyceric acid and then it is converted into glyceraldehyde 3-phosphate. You may think that if phosphates were supposed to be removed from 1,3-bisphosphoglyceric acid, then why did we add phosphate groups into phosphoglyceric acid to make 1,3-bisphosphoglyceric acid? So the answer here is that I told you that the dark reactions are catalyzed by enzyme. And you know, enzymes are very specific in nature. They are very specific for their substrate. So the enzyme which can act on 1,3-bisphosphoglyceric acid cannot act on phosphoglyceric acid. That's why we added phosphate groups to phosphoglyceric acid to make 1,3-bisphosphoglyceric acid. Got it? Now, the last step is regeneration of ribose bisphosphate. Now, how does this regeneration occurs? You saw that 6 molecules of G3P were produced in the process of reduction. Okay? Now, it contained 18 carbon and it was G3P. Now, what happens? That only one molecule of G3P is used as a net gain or a product of dark reaction. Only one molecule of G3P is used to make glucose or sucrose or starch or any other carbohydrate. The remaining 5 molecules are reused to regenerate ribose bisphosphate. Now, how the molecules are reused? That these molecules break from the center. Okay, these molecules break from here, it breaks from here and then it breaks from here. The molecules, after undergoing complex reactions, they rearrange themselves and make ribose phosphate. Okay, only one molecule of G3P is used to make glucose and all other stuff. So what happens? That these molecules, these first carbons arrange in this way, these second carbons arrange in this way, and these third carbons arrange in this way, and phosphate group is present on 5th carbon. So I told you that ribose, this is ribose phosphate. Ribose phosphate contain phosphate on the 5th carbon, but we have to generate ribose bisphosphate. It means that there must be 2 phosphate groups. 1,5 ribose bisphosphate, phosphate on carbon number 1 and carbon number 5. So the next step would be the addition of phosphate groups to this ribose phosphate, and phosphate will always be from ATP. As there are 3 molecules of ribose phosphate, so 3 ATP are used. These 3 ATP breaks down into 3 ADP and 3 inorganic phosphates. These inorganic phosphates attach to the carbon number 1 of ribose phosphate to make ribose bisphosphate. So it is the same structure, it is the same carbon dioxide acceptor which we studied in the very first step. So 3 molecules of 1,5-ribose bisphosphate which were used are now produced. That's why it is a cyclic process. Got it? So it is 1,5-ribose bisphosphate. It contains 15 carbon. Now this is the diagram which has been mentioned in your book, and this is the diagram which basically you have to learn. Firstly, look at the equation. We use 3 molecules of carbon dioxide. We use 6 NADPH and we used 9 ATP. The product was CH2O and whole 3. That means it is a triose in nature because it contains 3 carbon atoms and then 6 NADP. Here must be 6 NADP plus because NADP lost electrons too. Okay, and whenever there is loss of electrons, there is positive charge. So you will write it as 6 NADP plus and 9 ADP, 9 inorganic phosphates and 3 water molecules were produced, which were produced in the step of reduction. Now you can see here that input is 3 carbon dioxide. 3 carbon dioxide molecules add with the 3 molecules of 1,5-ribose bisphosphate in the presence of Rubisco to make firstly unstable 6 carbon compound, and then that 6 carbon compound breaks into 6 molecules of 3-phosphoglyceric acid or 3-phosphoglycerate. Now these 3-phosphoglyceric acid adds phosphate into it to make 1,3-bisphosphoglycerate, and in this step ATP, 6 molecules of ATP break down into 6 molecules of ADP and 6 inorganic phosphates. Now 1,3-bisphosphoglyceric acid or 1,3-bisphosphoglycerate removes 6 inorganic phosphates from it, adds 6 electrons from NADPH and makes glyceraldehyde 3-phosphate. This is the step from where the hydrogens are obtained from NADPH and oxygen from carbon dioxide, and the product is water. Now glyceraldehyde 3-phosphate contains, we have got 6 molecules of glyceraldehyde 3-phosphate, out of which only 1 molecule goes to make sugar or glucose or sucrose or starch or any other thing. So the net gain of the dark reaction is only 1 molecule of glyceraldehyde 3-phosphate. The other 5 molecules of glyceraldehyde 3-phosphate then undergo complex reactions to make ribulose phosphate. Okay, rearrange themselves to make ribulose phosphate, and this ribulose phosphate now adds 3 phosphates by breaking 3 ATP molecules, and then it makes 1,5-ribulose bisphosphate, 3 molecules of 1,5-ribulose bisphosphate. Okay, so this was all about the dark reactions, and you have to remember this diagram. Do listen to the lecture again, practice the diagram again and again, and then you will remember the names and all the process. Okay, it's quite easy, no difficulty. Just listen to the lecture carefully and memorize it in a good manner, and then practice the diagram again and again. Now here is the summary of the reaction. First point is that carbon dioxide is reduced to, sorry, here I should mention triose. Okay, glucose is not produced in dark reactions. Glyceraldehyde 3-phosphate then combines to form the glucose and hexose and all of this stuff, but the product of dark reaction is triose. ADP and NADP plus is regenerated. Okay, ATP and NADPH are used, and ADP and NADP plus was produced. Now ribulose bisphosphate is regenerated because it's a cyclic process, and G3PO or triose is the product of Calvin cycle, and this glyceraldehyde 3-phosphate can also be called as phosphoglyceraldehyde. Got it? 3 carbon dioxide molecules were used. Okay, 9 ATP were used, and 6 NADPH were used. Do remember, 3 carbon dioxide, 9 ATP, and 6 NADPH. Here I should mention pH. So 6 NADPH were used. Got it? Now it's the processing time. Okay, now I have designed some questions from you, and you have to answer that questions. First question is, dash is the carbon dioxide acceptor and precursor to phosphoglyceric acid or phosphoglycerate. Now you all know that the carbon dioxide acceptor is 1,5-ribulose bisphosphate, and precursor means the substance which comes before the production of phosphoglyceric acid. So ribulose bisphosphate is the precursor to phosphoglyceric acid, because ribulose bisphosphate adds carbon dioxide and then phosphoglyceric acid or 3-phosphoglycerate is formed. Got it? So this was the first question. The second question is, what is the first stable compound formed in the Calvin cycle? Do remember that the first stable compound formed in the Calvin cycle is this phosphoglyceric acid or 3-phosphoglycerate, while the product is glyceraldehyde 3-phosphate. This is the most important MCQs. Calvin cycle is called as C3 pathway due to phosphoglyceric acid. Calvin cycle produces the product glyceraldehyde 3-phosphate. Got it? The next is net gain of Calvin cycle. So net gain of Calvin cycle is 1 molecule of G3P. Which steps used ATP? Now this is the very important question for MDCAT. You saw that ATP were used at 2 steps. First step was the conversion of this 3-phosphoglyceric acid into 1,3-bisphosphoglycerate. Phosphate group was attached on the carbon number 1. Okay, and 6 ATP were used for this process because 6 molecules were there of 3-phosphoglyceric acid and 6 molecules were of 1,3-bisphosphoglycerate. And the second step was the conversion of G3P into 1,5-ribulose bisphosphate. You saw that G3P had only phosphate group on the 3rd carbon, so we rearranged this G3P into 5 carbons, and then phosphate group was attached on the carbon number 1, and on this step 3 ATP molecules were used. Got it? Now which step required least ATP? So the step of regeneration, there are 2 steps on where ATP was required. First step is the reduction. This was in the process of reduction, and this was in the process of regeneration. So the step of regeneration required least ATP, where G3P was converted into ribulose bisphosphate and only 3 ATP were required in this step. Which step required NADPH? So we all know that NADPH was required when 1,3-bisphosphoglyceric acid converted into glyceraldehyde 3-phosphate. 6 NADPH were used over here. So do remember that firstly 3-ribose bisphosphate adds carbon dioxide to make phosphoglyceric acid. This phosphoglyceric acid adds 6 molecule of ATP to make 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate now remove phosphate groups to make G3P. This G3P is rearranged to make 1,5-ribose bisphosphate. Got it? And do remember one more point, that all the ATP and NADPH were used in which step? So ATP was used in 2 steps, reduction and regeneration, while all of the NADPH was used in the step of reduction. Got it? So students, this was all from my side. I would request you to please practice the diagram, read from the textbook again and again. This topic is very, very important, not only for FSC but also for MD-CAT students, and students find this topic so difficult. So listen to the lecture again and again, practice it again and again. I have tried my best to make this lecture simple for you. So if you have got the lecture, have a big smile on your face and be happy, make others happy. Like the video, share it with your friends and subscribe to my YouTube channel. See you in the next video. Allah Hafiz.